this video, I'm going to go over the distinctions between B cells and other antigen-presenting cells and the way that they process antigens, and also looking at MHC class II molecules or proteins. So the first distinction, although macrophages, dendritic cells, and B cells are all antigen-presenting cells, there are differences in the way that they process antigens. Macrophages and dendritic cells are phagocytes. And what they're going to do is they're going to ingest and they're going to kill pathogens that penetrate that first line of defense, such as your mucous membranes and your skin. And then they can process those antigens and they can put them on their surface via their MHC class II proteins. But they're phagocytes. They take in the pathogen, they break it apart. Well, B cells don't function as phagocytes, but they play a primary role in the production and secretion of antibodies. Remember, B cells differentiate into plasma cells. Remember that when B cells get activated, they're going to differentiate into plasma cells, and the plasma cells produce antibodies. So they don't act as phagocytes, and they produce antibodies, which macrophage and dendritic cells don't do. The second major difference is that macrophages and dendritic cells are going to recognize pathogens through nonspecific receptor interactions, such as using their toll-like receptors to bind to PAMPs. PAMPs are pathogen-associated molecular patterns. And you can have multiple different toll-like receptors on a macrophage or dendritic cell that will bind to a PAMP. And then that's going to stimulate it to bring it in by phagocytosis. They also are involved in opsonization with complement antibodies. So for example, here's a bacterium, and you have antibodies bound to that bacterium. Well, the stem part of the antibody here and here can bind to a receptor that's on a phagocyte, like a macrophage, and then that stimulates that macrophage to bring in that particular antigen or pathogen that it's attached to. So it's involved in opsonization. Whereas B cells are going to interact with their pathogens or their free antigens using specific B cell receptors. So there are specific B cell receptors, as shown here, that will bind to a very specific kind of antigen. And then once it's bound to that antigen, then that antigen can be brought in by what's called receptor-mediated endocytosis. Very specific, more specific than a toll-like receptor will be for a PAMP. Because you can have multiple toll-like receptors, but on a B cell, it's only going to have one B cell receptor on that cell. And there could be thousands of these identical B cell receptors, of course, on that cell. And then once it's brought in, it can be processed. The pathogen, antigens can be broken apart, and then it will be put onto its surface by a MHC complex. Remember, B cells can use MHC1 and MHC2, as all antigen-presenting cells can do, but it primarily uses MHC class II. Why? Because these activate helper T cells. And if you activate a helper T cell, then that will then cause this helper T cell, as you can see here, to release chemicals, like interleukins, which then activate the B cell to make multiple clones. And some of these clones are going to become plasma cells that produce antibodies, and others are going to become memory cells. So helper T cells are really important in the activation of B cells. And you want to make sure you note this. Now, B cells can be activated without helper T cells. However, their activation is greatly enhanced with helper T cell activation, because helper T cells are going to release different substances that are going to enhance the activation of B cells, become memory cells and plasma cells. Let's test your knowledge. Pause the video and select the term that makes the sentence correct. Here are the answers. That concludes this video, and let me know if you have any questions.